Hello guys, in this video I'll be discussing histone proteins. What are histones? Histones are these proteins that basically DNA wraps around. They're made up of H2A, H2B, whatever. There's a whole list of names, but that's not the point of this video is to name them. But there's a bunch of, basically there's 8 of them that start with like H1, H2, blah blah blah. Actually H1 is important because it's different. H1 is located not in the core. So there's something called a histone core. There's 8 of them. Basically, what happens is DNA wraps around these histones, and what you call DNA wrapped around histones is called a nucleosome. A nucleosome, right, is the DNA wrapped around the histones, and this is the DNA. This line that I'm drawing here, that's and then this is the histone core with this is the DNA right here, and this is H1. It's one of the histones, but it's different because it's not within the core with the other histones like H2A, H2B, whatever. What it does actually, H1, it condenses the DNA even further to a particular size, which you might want to know. I don't know if you don't have to know it, but 30 nanometer is what this does. So the job of the histones basically is they're able to condense DNA. So we have chromosomes, right? Those are like the most condensed DNA, right? Before the cell divides and pulls them apart on opposite poles, right? In order for mitosis to complete, right? But basically, we never need to make a protein, as I talked a little bit about in my other video, is you need to decondense these. But what's necessary is that you need to create modifications to these histones. These histones are going to have these tails, right? These histone tails can be modified by various chemical groups to basically coil the DNA up or uncoil it. And there could be several modifications. For instance, you could have methylation. I'm just going to write CH3 for methylation. Phosphorylation. Acetylation. I'm going to draw an acetyl group because it might be easier than to write it out. Acetylation. And then deacetylation. I'm just going to write minus for deacetylation. So methylation, phosphorylation, acetylation, and then deacetylation. Deacetylation meaning, once again, to remove the acetyl group. These have different there could be exceptions, but generally speaking, when you the methylation tends to be a silencing thing, meaning that you're shutting down genes, and phosphorylation, that I'm not going to speak on because I think it's a little bit more complicated. But acetylation tends to decondense the DNA. Deacetylation, meaning removal of the acetyl group, tends to condense the DNA. And the way this works is these histone tails are typically rich in basic amino acids. Your basic amino acids are like arginine, lysine, all right? And lysine is actually not L, it's K. That's the one-letter abbreviation. Right? And these basic amino acids under physiological pH have a plus charge, right? And you know your DNA, I'm going to just draw a double helix here, right? Is made up of a sugar phosphate backbone, right? Of a phosphodiester bond, but the phosphate in the DNA's backbone has a negative charge, right? And we know that plus and minus charges attract, right? So